Hello and welcome. In this presentation, you will be introduced to culture media for growing animal cells in vitro, the basic components that make up a cell culture medium, and the types of culture media. An animal cell culture medium is a liquid or a gel that supports cell survival and proliferation, as well as cellular functions. The quality of the medium directly affects the results of a cell culture or your research, so the culture medium is the most important factor for culturing animal cells and tissues. The role of a cell culture medium is to provide an environment that is similar to our body or the body of the animal from which the cells were obtained originally. Cells get all the required nutrients like amino acids, salts, and glucose from the culture medium. All the essential amino acids are required by cultured cells. Also, other non-essential amino acids are added along with the essential amino acids just in case there is any loss by leakage into the medium or in case a particular cell type may not be able to make them. Inorganic salts are also essential components of a cultured medium, since they are the nutritional precursors for macromolecules and regulators of intracellular charge. Divalent cations like calcium ions are required for cell adhesion and in signal transduction. They can also influence whether cells will proliferate or differentiate. Sodium, potassium, and chloride ions regulate membrane potential, while sulfate, phosphate, and bicarbonate ions are anions required by the cell matrix. The culture medium also provides glucose as the main source of energy for the cells. Growth factors and hormones. Growth factors and hormones are supplemented in the medium to induce cell proliferation, differentiation, migration, secretion, and other cellular functions. Vitamins are also supplemented in the medium to ensure cell survival and growth. Like amino acids, vitamin requirements depend on the cell line originally used in the development of the medium. Also, various compounds including proteins, peptides, nucleosides, citric acid cycle intermediates, pyruvate, and lipids are found in complex medium as organic supplements. These substances help in maintaining certain specialized cell lines. Antibiotics are also added to the growth medium to reduce the frequency of contamination in the culture. pH. The culture medium also provides the right pH for optimum cell growth. Most mammalian cell lines grow well at pH 7.4. Phenol red is used as a pH indicator in the culture medium, and it imparts a red color to the culture medium at pH 7.4, becomes orange at pH 7, yellow at pH 6.5, and lemon yellow when the pH falls below 6.5. More pink at pH 7.6, and it turns purple at pH 7.8. Buffers are used to counter the rise or fall of pH, which may be attributed to the evolution of carbon dioxide by the growing cells. Culture medium also provides optimum temperature for the cell culture when incubated at the right temperature, especially for warm-blooded animal cells. This culture temperature is determined by the body temperature of the animal from which the cells were obtained, providing the right oxygen tension. While most cells require oxygen for respiration in vivo, cultured cells often rely on glycolysis and may be anaerobic. So high oxygen tension leads to increase in the level of free radicals in the medium. Cells only use dissolved oxygen, and so providing the correct oxygen tension is difficult, since it means fulfilling the respiratory requirement and avoiding toxicity at the same time. The cell culture medium also provides the right osmotic pressure. In practice, osmolalities between 260 osmoles per kilogram and 320 osmoles per kilogram are quite acceptable for most cells. Certain culture media are also supplemented with serum for its growth promoting and protective factors to cell cultures. Types of media. There are natural media and synthetic or artificial media. Natural media. These are media containing naturally occurring ingredients. In spite of a vast use of chemically defined media in animal tissue culture, it is still necessary in most culture undertakings to depend on naturally occurring substances. Blood plasma. Tissue culture was first conducted by Ross Harrison in 1907 in clotted frog lymph. Burroughs in 1910 substituted a coagulum prepared from chicken plasma. After many years, it was found that plasma provided a complete nutrient in which cells could survive and multiply slowly for extended periods under in vivo-like conditions. So plasma is still being used for conditioning the medium and to provide a nutritive substrate and a supporting structure for many types of cultures, just like it provides a matrix for new cells during repair of injured tissues in the body. It is also used to provide a means of conditioning for better attachment of cultured cells to substrate and to provide a means of protecting the cells and tissues from trauma during subculturing. Blood serum. Blood serum, which is plasma minus the fibrinogen, is the most commonly used biological fluid in the culture medium and is obtained from human adult blood, placental cord blood, horse blood, or calf blood. Besides serum, other biological fluids used as natural media are amniotic fluid, the lymph, tissue extracts, plasma clots, and insect hemolymph. Balanced salt solutions. Although natural media are convenient, their exact constitution is not known clearly and they cannot be reproduced with the exact composition. For this reason, synthetic media have been designed for a variety of tissues and purposes. Balanced salt solutions form the basis of these media. So a balanced salt solution is composed of inorganic salts of mainly sodium, potassium, calcium, magnesium, and chloride. Balanced salt solution has a defined osmotic pressure and pH, which is obtained by a combination of the inorganic ions, salt, and glucose. Balanced salt solution may be used as a washing or a dissection medium for immediate survival of cells and tissue explants. Also, balanced salt solution may be supplemented with amino acids, vitamins, serum, proteins, growth hormones for longer survival of cells, so it's used as a diluent for making complete media. There are different balanced salt solutions with varying compositions, and examples are Earl's balanced salt solution, Hank's balanced salt solution, Delbaker's balanced salt solution. They form the basis of many complete media, like the Eagle's minimum essential medium with Hank's balanced salt solution, or the Eagle's minimum essential medium with Earl's balanced salt solution. Basal media. In 1912, Alexis Carrel demonstrated the possibility of long-term cultivation of connective tissues of chick fetus cells in a medium supplemented with embryonic tissue extracts. In 1955, Harry Eagle studied the minimum necessary amounts of low molecular weight components that are required by mouse L cells and HeLa cells. He used a balanced salt solution with the addition of dialyzed serum and found that 13 amino acids and 8 vitamins are necessary. Eagle then developed the minimum essential medium MEM which is composed of the minimum essential components that he identified. That is glucose, 6 inorganic salts, 13 amino acids, 8 water-soluble vitamins, and dialyzed serum. Several improvements were made to the minimum essential medium on the basis of the cell type, the origin, that is the origin of the animal species, and the purpose of the culturing, like Dalbecko's modified Eagle's medium, Iskov's modified Dalbecko's medium, Ham's F10, RPM I1640, etc. Now let's discuss serum containing media. Commonly used basal media like DMEM, RPMI, etc. are supplemented with serum because serum contains growth factors which promote cell proliferation and adhesion factors and antitrypsin activity which promote cell attachment. Serum is also a source of minerals, lipids, and hormones, and sera mostly used are calf serum, fetal bovine serum, horse serum, and human serum. Some examples of media supplemented with serum are the Ham's F12, which is supplemented with fetal bovine serum. This is used for culturing chondrocytes. Then we have the Eagle's minimum essential medium supplemented with calf serum and used for culturing HeLa cells. The RPM I1640 supplemented with fetal bovine serum used for culturing memory epithelial cells. Then we have a combination of DMEM and F12, which is supplemented with both fetal bovine and horse serum, and this is used for culturing skeletal muscle cells. On the other hand, using serum in the medium has a number of disadvantages. Physiological variability. The major constituents of serum like albumin and transferrin are known, but serum also contains a wide range of minor components that may have a considerable effect on cell growth. These components include peptide growth factors, hormones, minerals, and lipids. The concentrations and actions of these minor components have not yet been fully determined. Short shelf life and variable consistency. Serum deteriorates very soon and may last as long as 1 year. Also, serum varies from batch to batch, so it must then be replaced by another batch that may be selected as similar but will never be identical to the first batch. Quality control. Changing serum batches requires extensive testing to ensure that the replacement is as close as possible to the previous batch. This can be tedious as it involves several tests for growth, plating efficiency, and special functions, and a number of different cell lines. Specificity. If more than one cell type is used, each type may require a different batch of serum so that several batches may be kept on reserve simultaneously. Co-culturing different cell types will be even more difficult if serum is used in the growth media. Availability. Sometimes the supply of serum is restricted due to drought or the spread of disease among cattle or due to economic and political reasons. This can create problems at any time, thus restricting the amount of serum available. And downstream processing. The presence of serum in the growth medium creates a major obstacle to purification and recovery of cell products by the cell cultures. Risk of contamination. Serum is frequently contaminated with virus, which is an unwanted factor, even if it may be harmless to the cell culture. Cost. Serum supplementation increases the cost of the medium because serum constitutes a major part of the cost of a bottle of medium. Apart from its growth-promoting activity, serum also contains certain factors that may be growth-inhibiting to some cell types. Next is serum-free media. The total omission of serum and proteins is thought to have begun in 1946 by Philip R. White. He developed a chemically defined medium that was composed of glucose, inorganic salts, amino acids, iron, vitamins, and glutathione, which is a protein-free media. So serum substitutes like growth factors, adhesion factors, hormones, mineral nutrients, etc. are added to the existing media like RPMI 1640. Media combined like HAMS 12 with Dalbeco's modified Eagles medium with supplements like selenium, transferrin, insulin, etc. to develop serum-free media. The need to standardize media among laboratories and to remove undesirable, uncontrolled, and often contaminated natural products, particularly serum, from the culture and its cell products led to the development of more complex and chemically defined media. Examples are the M199, the CMRL 1066, HAMS F10, and HAMS F12 for Chinese hamster ovarian cells. Then you have the MCDB series of media, Sato's DMEM, F12 based media, and others based on RPMI 1640. The advantages of using serum-free media are the selectivity or specificity of a particular cell type, and the ability to regulate the proliferation and differentiation of cultured cells. The disadvantages of using serum-free media are the multiplicity, that is, each cell type appears to have different requirements. So while the specificity of a serum-free media may be advantageous for isolating specific cell types, it is a problem for culturing several cell lines of different origins. Selectivity. Cells at different stages of development may require different media formulations, especially in the growth factor and cytokine composition. Then we have the reagent purity issue. The removal of serum also removes the protective and detoxifying action of some serum proteins. So the removal of serum also requires a higher degree of cleanliness and aseptic conditions of glassware, reagents, water, etc. Slower cell proliferation and growth. And then it's more expensive. So the availability of properly controlled serum-free media is limited, and so the products are often more expensive than conventional media. So that's all we'll be learning for today. Thank you.